Okay, I think we can go ahead and get started now. I hope you can all hear me. I've been having some issues with the microphone, so we'll see what happens. And if you can't hear me, I'm sure somebody will point it out to me. So
we can hear you now.
Okay, good. That's all I need to know. So, okay, we're going to go ahead and move into the rest of the metabolism chapter. I'd like to wrap that up today if we can, and move forward. I've been trying to go at a reasonable pace because this is not the easiest material to grasp. And you know, it's one of those things that you need to understand how we get energy in our cells. But no one's ever going to ask you to explain exactly what happens in the mitochondria as far as cellular respiration goes, except me in a test. But in a clinical setting, you're never going to be challenged about that. However, there is a good reason to understand exactly where we use oxygen at the cells. So that's what hopefully you're going to have a good understanding of this when we wrap this up today. So I'm going to go ahead and open this up and go into chapter 24. And we talked about anabolic and catabolic reactions a little bit. And we were just about, here we go. The term cellular respiration. Okay, cellular respiration does not involve breathing. I mean, ultimately, it uses oxygen, but it has nothing to do with breathing. You know, your airway opening up and your lungs inflating and deflating and stuff like that. Cellular respiration is the term we use for generating ATP generating ATP inside our cells. Now, there are three types of chemical reactions that take place. One is called glycolysis. Glycolysis is a bit of a free ride because it's the first time that we can do something to the glucose molecule. Like we've been saying all along, that glucose is what we use for energy. It's our energy source, and we strip off electrons from glucose. We break the covalent bonds, and we take the potential energy from those electrons, and we use it, and we do. This first process is known as glycolysis. We don't break a lot of bonds here. We don't steal a lot of electrons, but we take some, and we get some of the potential energy. Remember that. How do we know there's potential energy in glucose? Because it's already been measured. You know, so the calorie content in a gram of glucose in a food item that we eat, like an apple or a potato or a Snickers bar or something like that. The calories represent the potential energy. That's already been predetermined for us. We don't have to go and look at a Snickers bar and say, well, let me quickly determine there are how many glucose molecules in here and how many hydrogens do I have and how many carbons do I have and how many electrons could I possibly have here from all those carbons and hydrogens and how much potential energy is available. It's already been determined. If it says 180 calories in the label, that's a measure. That's the measure of potential energy in the Snickers bar, or 60 calories on an apple. And we want to get some of that energy. That total for any food item is the sum of all the potential energy stored in that food, like in an apple. The pulp of the apple, the starchy pulp of the apple. How many calories are available in that to us? How much that apple represents stored energy? How much of that can we use? That's all potential energy. So like a battery. You know, battery is stored energy. An apple is a natural battery, if you will. Now, the three processes that we use to get some of this energy into a form that we can use, because we can't take that energy directly out of the apple and use it. We can't process that much energy coming loose all at once. We would literally likely explode. We don't like that. That's not something we want to do. So we have to break it down slowly. We have to release the energy slowly. If you put gasoline in your tank or diesel in your tank, whatever fuel you're using, you wouldn't want all the energy in the gasoline to reach your engine at exactly the same time. There's a lot of stored energy in that gasoline, a lot of valence electrons in that gasoline. You don't want all that energy released at exactly the same time in your engine. Your engine would explode. Your car would go very fast for a very, very short period of time. But it's not, we don't want that. We want to have a nice controlled release of gasoline energy in our car when we drive it, so it lasts longer and it doesn't explode. That's why we have to have a controlled process for breaking down the stored energy, or making the stored energy in an apple or a Snickers bar or a Pepsi, release in a way that we can use it. So the first step is called glycolysis. Glycolysis takes place in the cytoplasm. It's the only process of these three steps that takes place in the cytoplasm. That's an important point to remember, that glycolysis only occurs in the cytoplasm. I can't emphasize that point enough, if you get my drift. The second stage is called the citric acid cycle, or what we usually call the Krebs cycle. Krebs was a biochemist that discovered this pattern of transfer of energy inside the cells, and so it's been named after him for years and years and years. But it's also called the citric acid cycle because we use one of the components of the cycle is citric acid, and I'll show you that here in a minute. And we get a little energy from the citric acid cycle too. We can grab some electrons in the citric acid cycle, the Krebs cycle. It takes place inside the mitochondria. The big payoff that gives us a lot of ATP, because glycolysis gives us a little ATP, Krebs cycle gives us a little ATP, electron transport gives us a lot of ATP. And it's not so much, we're going to see here, it really is the movement of the electrons that's going to do this for us. So electron transport, which takes place inside the membrane of the inner membrane of the mitochondria. Electron transport, the inner membrane of the mitochondria, is where we get our big energy payoff. And the end result of cellular respiration is always going to be carbon dioxide and water and ATP for us to use to transport energy around and the release of heat. 60% of the energy stored in the food that we eat comes off as body heat. We don't use it. It keeps us warm. It keeps us at 98.6 or 37 degrees Celsius. And our bodies are designed to operate at 37 degrees. But that 60% that comes off as heat is energy that's not usable, because that heat radiates away from us and we can't use it anymore. It's not lost, it's not destroyed, it's just not available. So carbon dioxide, water, ATP, and heat, those are the products here. The carbon dioxide that we're breathing out right now is a result of cellular respiration inside our mitochondria. Now, these reactions occur without oxygen and with oxygen. We have chemical reactions called anaerobic reactions. Anaerobic without the AN in front of aerobic. Aerobic means oxygen related. Anaerobic means without oxygen, and it makes a little ATP. You can deplete all the stored oxygen in your muscle cells, and the cells will still function. They'll still get their energy. They won't get very much energy. They'll continue to function the energy levels will be at a much reduced rate, but they'll still function. But it's entirely possible for us to deplete all of the stored oxygen in skeletal muscle. We call it oxygen debt. We store that oxygen in the respiratory pigment called myoglobin. Myoglobin is the pigment in muscle that makes it look red. You down and look at the meat counter at Food City. You go into the beef counter and you look at all the roasts and the steaks and the ground beef and stuff like that, and it's all red. It's red not because of blood, it's red because of the presence of myoglobin. Myoglobin holds oxygen, and myoglobin is related to hemoglobin in that it's a respiratory pigment, just like hemoglobin is. It holds oxygen for us in the muscle cells, and it gets bright red when it's full of oxygen. And when it's given up oxygen, it turns brown. Now, that does not mean that the meat has gone bad, by the way. Meat that has gone from bright red to sort of a brown appearance is still perfectly fine. It just doesn't look as pretty. Now, the anaerobic portion of cellular respiration will continue to function in the absence of oxygen. Even though we don't have any oxygen available, the cells are still going to be making a little ATP. If you've depleted, you ran a marathon. The Boston Marathon was this week, and it's 26 miles and some odd yards to go with it. And at the end of the race, most of the runners were in oxygen debt. They depleted all the oxygen in their muscles, but their muscles were still working because they were running anaerobically. We prefer aerobic reactions. We prefer to use oxygen, but we produce a lot of ATP when we use oxygen in our muscles. But it's okay if we don't use oxygen. Okay, one of the concepts that's difficult, I was talking about this, this is where I stopped last time. Oxidation-reduction reactions. Oxidation, it is essentially the loss of an electron. When something is oxidized, it loses electrons, it loses energy. It says here, the loss of a hydrogen. Okay, every time you lose an electron, you lose a hydrogen with it, because electrons are negatively charged and they won't go anywhere unless they take their best friend along, and their best friend is a hydrogen ion. So the electron and the hydrogen ion leave together. When we rip apart a covalent bond and we release electrons, we're releasing hydrogen ions with it. So during oxidation, we give up electrons. Now, if you light a candle, take a match and light a candle, or a lighter, light a candle. The candle's going to burn. The wick is going to burn. It's using the wax of the candle, the paraffin, as its fuel. And you're breaking the covalent bonds that hold the wax together. And you're releasing heat and you're releasing light. Those electrons, when you break the covalent bond in the paraffin and release the electrons, it gives off heat and light, and it consumes the wax. The wax is broken apart. The heat comes off. The heat's not destroyed. The heat spreads out to where it's no longer useful. The light spreads out to where it's no longer useful. And then at some point, the further you are away from a light source, you're unable to see it. You get far enough away, you can't see the light anymore. You may not see, if you're a mile away from someone holding a candle that's burning, you likely won't be able to, and it was a totally flat area, you likely wouldn't see the flicker of the candle. You definitely wouldn't feel any heat from it. I mean, consider a fire pit or a fireplace or a grill. It's very warm up near the front of the grill or near the fire pit or near the fireplace, but your backside is cold. If you're outside of the fire pit, your front's warm and your backside's cold. The heat doesn't go very far. Same with a fireplace. The heat feels good around the front of the fireplace, but you go into another room and you don't feel that heat anymore. It has spread out already. Anyway, I don't want to get down a rabbit hole yet. Oxidation is giving up electrons and giving up energy. Oxidation releases electrons, and in doing so, it releases the potential energy. When we burn anything, it's oxidation, and we release electrons. When a shovel sits out all winter and rusts, that's oxidation. As the shovel's getting weaker, it is losing energy as it's giving off its electrons. It's losing energy. It's rusting. It's forming iron oxide, which is a very weak substance. That's why things turn brown. That's iron oxide. Anything that is undergoing oxidation gives up electrons, and there's no difference between a shovel rusting and burning wood in a fire pit, or burning gasoline in your engine, or lighting a firecracker, except time. Rusting is slow oxidation. Lighting a firecracker is rapid oxidation, but there is no difference in there other than the speed of the reaction. Slow oxidation is rusting, rapid oxidation is a firecracker. Lighting a match is fairly rapid oxidation, but it's not as fast as the explosion from a firecracker. So it's all about, every one of those, or eating an apple. That's somewhere a little faster, breaking down the energy in an apple is a little faster than a rusty shovel, but it's still pretty slow. We don't want that apple to release all of its energy at once. That's what happens in a firecracker. It releases everything at once. We don't want a candle to release all of its energy at once. It burns slowly. Otherwise, it would flare and be gone in one bright flash. We don't want that. Can you imagine if you put a shovel outside and it started to rust and suddenly it just flashed and disappeared because it had rapid oxidation? Now, there's another term here called reduction. Oxidation is giving up electrons and releasing energy. Reduction means something has to gain those electrons and gain that energy, because the universe has to stay balanced. If we give up an electron, we have to have something take it. Something has to accept the electron. It just doesn't run randomly out, I'm free, running free from the shovel. It doesn't do that. It has to be grabbed by something. And so that something grabs electrons. Now, a chemist will tell you that when a substance grabs a lot of electrons, it has become reduced. Now, to most of us, reduced means it has shrunk. Now, in chemist talk, reduced means that the number of positive charges have been reduced because there's more electrons. Okay, that's what reduction means. Reduction means gaining electrons and gaining the energy. So usually what happens in oxidation and reduction is that some substance gives off an electron and releases energy, and oxygen grabs those electrons and grabs the hydrogens that travel with it, and it forms water. What is water? H2O, 2 hydrogens and 1 oxygen. The oxygen molecule would love to pick up, it's not, it would love to, an oxygen atom would love to pick up 2 more electrons to be stable in its valence shell, to have that valence shell filled up. Hydrogens want to be filled up too. So hydrogens and oxygens readily combine. And what the consequences of an oxidation-reduction reaction is usually the breakdown of a substance, the release of its electrons, and the formation of water. We call these redox reactions. The donor, the substance that gives up the electrons, is the reducing agent. The acceptor is the oxidizing agent. Now this is confusing because the donor, if you're burning a piece of wood, it's giving up its electrons. It's the reducing agent because it's giving electrons to the atmosphere, which is forming water vapor. The electron acceptor is the oxidizing agent. Now this might make a little more sense because if you don't have oxygen, you can't burn something. You can't have a fire without oxygen. Things don't burn in outer space or on the moon. You gotta have oxygen. If you remove the oxygen, then the fire would go out. We smother a fire with a fire extinguisher, and the fire goes out. We put water on a fire, and we go down below the combustion point. We eliminate the use of the oxygen. So electron donors are the reducing agent, electron acceptors are the oxidizing agent. For oxidation to occur, we've got to have something that grabs the electrons. Okay, so that's your chemistry lesson there. Now this little illustration may be helpful. If this is a piece of wood or a candle, it's got lots of these electrons here represent their stored energy. When it becomes oxidized, it has lost those electrons. Before it can get oxidized, something has to be able to grab those electrons. So we're going to burn that piece of wood, and the atmospheric air with lots of oxygen is going to grab those electrons and grab those hydrogens. If we are burning fuel, if we are doing anything like that, we're leaving our shovel outside, we're going to give up those electrons here, and it's going to get transferred to oxygen. Except, what if you can't transfer the electrons? What if you put a coat of Rust-Oleum over your shovel? Rust-Oleum is that it seals the metal. It keeps the oxygen out. Your shovel doesn't rust. You paint the metal part of your shovel with Rust-Oleum, you seal it, and no rust. The oxygen can't get there. Oxidation only works if we have something to grab the electrons. Now, usually oxygen is the oxidizing agent. Usually oxygen is what's going to grab electrons. Usually. So anyway, that's a redox reaction. Now in our cells, we do have redox reactions because we are removing electrons from fuel. The food that we eat, the carbohydrates, lipids, things like that. We take electrons from the glucose and we transfer them to another molecule. So when we lose the electrons, it's called, it's still oxidation. When you're breaking down that Snickers bar and you are releasing the electrons, that's oxidation. And something has to grab those electrons. And so that whatever is grabbing those electrons is becoming reduced, because the definition here from Chemist Talk is that adding electrons is called reduction because this reduces the number of positive charges in the accepting molecule. So when oxygen grabs those extra electrons coming off of the glucose, oxygen gets reduced. It gets more electrons. Remember, oxygen has atomic number of 8, 2 electrons in its first shell, 6 in its second shell. It has room for 2 more. When it grabs those 2 electrons and the hydrogens to make water, the oxygen has become reduced and has picked up more electrons. So, and our bodies do this all the time. We transfer the energy from glucose to the third phosphate of the ATP. But that's only a temporary move, because that only lasts for 7 seconds. We don't destroy those electrons. The electrons will usually end up being grabbed by oxygen. Now this is an oxidation-reduction reaction. Every time you light a candle or burn a fire or start your car, that's an oxidation-reduction reaction. Can't light a candle without oxygen present. Oxygen is being consumed here. Carbon dioxide and water are coming off. The end result of an oxidation-reduction reaction is carbon dioxide and water. Guess what? We breathe out carbon dioxide and water vapor. Now, if you were to put and take a beaker and cover these candles, the candles would go out. And it's not because the oxygen's missing, but it's because carbon dioxide is present. If you take a candle inside a beaker, you have a candle inside a small beaker that's burning. You take a larger beaker and set it on top of that. Two things are going to happen. The candle's going to go out, and you're going to see a band of moisture, water, condense on the side around the inside of the beaker. Because what's coming off of this? Carbon dioxide and water. The oxygen doesn't, the candles don't use up all the oxygen. They're producing so much carbon dioxide that it displaces the oxygen. Carbon dioxide is heavier than oxygen. It pushes the oxygen out and drops down below. Oxygen is at the top of the beaker and carbon dioxide is down where the candles are, and the candles can't burn in the presence of carbon dioxide. So every time you're burning something, you're giving off carbon dioxide and water and heat and light. You are ripping electrons away from the fuel. You are breaking covalent bonds. Our bodies do this in a very controlled manner because we don't want to explode. Because if we released all the energy in a molecule of glucose at once, it would probably be unpleasant. We know what rapid oxidation can do. We light a match, that's rapid oxidation. We light a firecracker, that's even more rapid oxidation. Burning wood in a fire pit is sort of rapid oxidation. It's a lot faster than a shovel rusting, but we don't release all the energy at one time. That's not a good idea. We break down our glucose slowly. We use enzymes so that we can catalyze every step along the way. We strip away the electrons. We pull their hydrogens with them, because hydrogens are positively charged and electrons are negatively charged, because the hydrogen ion is just a proton. Glucose has 6 carbons, 12 hydrogens, and 6 oxygens. Every time we pull off an electron, one of the hydrogens is going to come with it. Now, in the initial step, we don't immediately transfer those hydrogens to oxygen. We're going to get there, but we use intermediate steps to transfer those hydrogens to oxygen. When we oxidize a Snickers bar, we release the electrons, we release the hydrogens to come with it, and they're ultimately going to end up with oxygen. But if they were to go immediately to the oxygen, it would be briefly exciting. So we go through a series of intermediate steps that are a little slower so that we don't release everything at once. And we do have something that accepts the hydrogens temporarily. They are the hydrogen carriers. They are the molecules NAD and FAD. And they can, when they carry a hydrogen, they're called NADH or FADH2. So this is what happens uncontrolled. This is uncontrolled oxidation. This is rapid oxidation. This is real-time video. And there's a lot of stored energy in the Hindenburg's fuel. I mean, or filled with gas bags with hydrogen, because hydrogen is lighter than atmospheric air, and so the Zeppelin here could float. It could float in the atmosphere. However, if you triggered oxidation, if a spark hit that hydrogen gas, which was very flammable, you would have rapid oxidation and form a lot of carbon dioxide in the water. And so this is what we don't want to happen in our bodies. This was not a step-by-step process. This was a rapid process. So, okay, now we have to slow things down in our bodies, we have to use intermediate steps. And we use something to accept the hydrogens and the electrons. We use NAD+, it has a positive charge, and FAD, nicotinamide adenine dinucleotide, which is why we call it NAD+, and flavin adenine dinucleotide, FAD. They're mouthfuls, but this is what we use to slow the process down. We slow the process down. We don't explode when we eat a Snickers bar or an apple, and we can capture the energy. So the end result is, with aerobic cellular respiration, we will ultimately get those hydrogens that we're pulling off. Remember, hydrogens, when I say hydrogens, I also mean electrons. So electrons and hydrogens are BFFs. They travel together, and we ultimately are going to get those hydrogens and those electrons from glucose to oxygen. We're going to be transferring hydrogen atoms along the way to get there. And when a hydrogen atom moves, it drags its electron with it. Or when an electron moves, it drags its hydrogen with it. Every time you release an electron, you're going to release a hydrogen too, and they're going to travel together. And the end result is to get the electrons and the hydrogens to oxygen. We want to do it at a little more controlled pace. So, and it is the valence electrons that are giving up their potential energy. Remember, the valence electrons have the greatest amount of potential energy. They're the ones in the farthest outer shell. I guess that's redundant. The valence shell is the outer shell. That has all the potential energy you're going to get from a molecule. If we release those electrons, we can take that potential energy with them. And the covalent bonds, when they're broken, will give off that energy, traveling with the electrons and traveling with the hydrogens. Something wants those electrons and hydrogens, and that something is oxygen. And we're going to get it there, just not immediately. It has to go through a series of other steps here. So now, the bottom line is that when we break the covalent bonds, that energy that is released can be used to make ATP. Yeah. ATP is nothing but a nucleotide, not a nucleotide. A nucleotide. It's a phosphate and a 5-carbon sugar, and instead of it's a base and a 5-carbon sugar and a phosphate. But instead of one phosphate, it has 3 phosphates: ATP adenosine triphosphate. Adenosine is the nitrogenous base. It has a 5-carbon sugar. It looks a lot like RNA, except that there are 3 phosphates on the end of it. Well, that 3rd phosphate is held onto the first 2 by a high energy bond. When that bond breaks, it releases energy. And then the phosphates, the phosphate is free, and the ADP, the adenosine diphosphate version, is free. Energy comes along again and puts them back together again and parks that energy at that bond. That's how we make ATP. We simply take energy and the phosphate and the ADP and glue it back together again. And energy is coming from the electrons from glucose. So here's your formula. Very simple formula to explain cellular respiration. C6H12O6, that's glucose, plus 6 oxygens will yield 6 carbon dioxides and 6 waters, and the release of energy in the form of ATP and then also heat. Glucose gets oxidized to carbon dioxide. It gives up 12 hydrogen atoms and 12 electrons. That forms CO2. Oxygen gains those electrons and those hydrogens and becomes water. Those 12 hydrogen atoms end up being attached to 6 oxygen molecules, 6 oxygen atoms. And we get 6 CO2s and 6 waters. Nothing special happens here. We haven't lost, the 6 carbons haven't been destroyed. The 12 hydrogens still show up attached to the oxygens as to form water molecules. The 6 oxygens right there with the CO2 and over here. Now, so what happens here is glucose ends up being converted, being oxidized to carbon dioxide with the release of a lot of energy. So how do we get the electrons off the glucose? Well, there are these enzymes called dehydrogenase enzymes, meaning they break the bonds and hold the hydrogens present. Dehydrogenase, it's an enzyme. And when something is, remember when we saw hydrogens being added to oils to make them solid, hydrogenation, we add hydrogens. Well, this is dehydrogenation. We're taking hydrogens away. These enzymes, dehydrogenase enzymes, will strip the hydrogens away from glucose and their electrons, and it will transfer those hydrogens to NAD plus, and along with their electrons. NAD plus accepts those electrons. So we have glucose over here with hydrogens, and in the presence of NAD plus, we are going to end up producing NADH plus an extra hydrogen out here. And that extra hydrogen wants to grab another electron. So protons are hydrogens. Hydrogens have positive charges. Electrons always take a hydrogen with them when they are ripped out of the covalent bond. They travel together. Now, our food contains lots of high energy substances. The atoms, the carbons, the hydrogens, and the protons and electrons in food contain lots of valence electrons with lots of potential energy. that potential energy from the hydrogens and the electrons is given to ATP. Now the hydrogens are low energy, the electrons are low energy, and they're looking for a home, and they find it in oxygen. So a molecule of glucose has a lot of potential energy. And when we're done with it, we transfer that all to ATP. The hydrogens and electrons have to go somewhere, and they are grabbed by the oxygen that's present. We form water, and we now have energy to use briefly. So food is broken down by the enzyme, the dehydrogenase enzyme, to strip off the hydrogens and electrons. NADH grabs those hydrogens and electrons temporarily, transfers them to the mitochondria, and then we use that energy in the mitochondria to make ATP. And now we have our worn out electrons and our worn out hydrogens, and the oxygen comes along and says, I'll take you home. And it forms water. Now, every ATP molecule has about 7 kilocalories of energy, 7.3 to be exact, 7.3 kilocalories of energy in each ATP molecule. And we make it two ways. Now, this is an important slide. Circle both number 1 and number 2 on here. Glycolysis uses what we call substrate level phosphorylation. Substrate means it's done by enzymes. Remember, the substrates are what go on the enzyme to, the substrates land on the active site of an enzyme, and a reaction occurs. This is where the dehydrogenase enzymes are working on the glucose to strip off the hydrogens and their electrons. Substrate level phosphorylation takes place in glucose. Substrate level phosphorylation, takes place in glycolysis on glucose. It doesn't use oxygen. Remember, glycolysis is oxygen free. It's anaerobic. Does not use oxygen. Substrate level phosphorylation, glycolysis only. Not a big payoff either. Oxidative phosphorylation takes place in the mitochondria in electron transport, because here we have to use oxygen to grab the electrons when we're done with it. So the two types of ATP production are substrate level phosphorylation in glycolysis and oxidative phosphorylation in electron transport. Make sure you know what that means. Make sure you know the difference between the two. Granted, no one's ever going to run up to you on the street and says, hey, what about oxidative phosphorylation? No. However, I can tell you for testing purposes, you're going to need to know it. So anyway, substrate level phosphorylation. The phosphate, the substrates, the portions of the glucose molecule land on the active sites of the enzymes, and we strip off those electrons, we strip off the energy, we strip off the hydrogens, and we can use that energy to make ATP. We put that high energy coming off the high energy from the covalent bonds that we're breaking can go right into the ATP. Every ATP molecule that gets formed gives you 7.3 kilocalories of energy. It sounds like it's a pretty good deal. This is direct transfer to the ATP. We say it's phosphorylated because we are taking the third phosphate and jamming it onto the ADP. Notice here in the illustration, adenosine diphosphate. we're taking the energy from the substrates and using that to force the third phosphate back on. This is a high energy bond. Substrate level phosphorylation, glycolysis, and arguably the Krebs cycle too. Does not give you a big payoff on ATP there. Oxidative phosphorylation is different. Takes place only in the mitochondria, uses electron transport, actually moves the electrons around. We essentially are going to take all these hydrogens that are coming off of the food, off the glucose. Every time we strip an electron, we grab a hydrogen. Remember, all these hydrogens are pumped across the membrane of the mitochondria to the inside inner membrane space and then allowed to diffuse back through. And that actually turns a pump in the membrane of the mitochondria, and that generates ATP. I just want to show you all this here. So, okay, this is oxidative phosphorylation. We're looking at this is the inside of the mitochondria. This is the mitochondrial membrane right here. Now, the energy from the Snickers bar or the apple or the potato that we ate, the energy that we have in those high energy electrons is used to pump hydrogens. The hydrogens that are traveling with those electrons in the mitochondria are pumped across this inner membrane. This is the inner membrane somewhere way out above. This is the outer membrane. There's a space in here called the inner membrane space. And we pump hydrogens into that space. And we put so many hydrogens out there that there are more hydrogens here than there are down here, and they want to come back in. They want to reach equilibrium. The only place that they can come through is through this structure here called an ATP synthase. It's an enzyme embedded in the membrane that makes ATP. And as these hydrogens come through here, they cause this thing to rotate, and it mechanically forces third phosphates onto all the ADP that's around, and we make ATP. And these hydrogens then come down here. So, now let's back up and take it nice and slow here and try to keep it as simple as we can. Of course, some of you might say that most of you are saying that we're way past the point of being simple, but I want to try. Carbohydrate metabolism, the breakdown of the starches, the sugars. We have to oxidize glucose. We are going to rip electrons off the glucose and their hydrogens. So glucose, C6H12O6, in the presence of 6 oxygens, will give you 6 waters, 6 carbon dioxides, and 36 ATP molecules. And 36 ATP molecules times 7.3 kilocalories is somewhere in the neighborhood, I think, of around 280 kilocalories of energy that we can use. It's a lot of energy. But that's only 40% of what was available. The other 60% of the energy in a glucose molecule ends up coming off as heat. This only represents 40% of the energy available in glucose. The rest of it comes off as heat. It's lost to us as usable energy. But we do have 36 ATPs to do things with. And that gives us the energy that we use all the time. Now, there are 3 ways we get this ATP: glycolysis, the Krebs cycle, and electron transport and oxidative phosphorylation. This picture makes perfect sense. No, this is a very busy slide, but it does show you where everything's happening. Glycolysis takes place in the cytoplasm. The Krebs cycle takes place in the matrix of the mitochondria, and electron transport takes place in the inner membrane. This is the inner membrane in here. Electron transport takes place in the inner membrane. Electron transport is the big payoff step. Glycolysis and Krebs cycle give you a little energy, give you a little ATP. Their role is much more important in the harvesting of electrons and hydrogens than it is in producing ATP. Electron transport is the big ATP producer. So glycolysis in the cytoplasm, substrate level phosphorylation. Remember, we use the dehydrogenase enzymes, and we strip hydrogens and electrons off the glucose, and we transfer that energy with the enzyme to the third phosphate to form ATP. It's an anaerobic process, doesn't use oxygen. The end result of glycolysis, and this is an important thing to remember, the end result of glycolysis takes a 6-carbon glucose and gives you 2 3-carbon pyruvic acids. That's what glycolysis does. It gives you 4 ATPs and 2 pyruvic acids. Doesn't do a whole lot for us, but it will keep the cells functioning, even if there's no oxygen present. So the end result of glycolysis, 2 pyruvic acid molecules. Pyruvic acid, each one is a 3-carbon compound. All we've done is we sliced the glucose molecule in half. 3 carbons and 3 carbons. If we have oxygen present, the pyruvic acid goes into the mitochondria. If it isn't present, it's converted to, if oxygen isn't present, we convert it to lactic acid. And if we've depleted all the oxygen in our muscle cells, we start building up lactic acid. Lactic acid makes our muscles hurt. If you have exercised so strenuously, you run a marathon, you've hiked up Mount LeConte and back down again, back down. You are likely, well, it's a reasonable assumption that you are probably experiencing oxygen debt in doing so. Without oxygen, you are running off of glycolysis alone in your muscle cells, and you're generating pyruvic acid, but there is no oxygen left to go into the mitochondria. So you're producing lactic acid, and lactic acid makes your muscles sore. So you have an accumulation of lactic acid, and you have oxygen debt, and your legs hurt, and they really hurt the next day. And it usually takes about 24 hours for the lactic acid to go away and for the oxygen debt to be paid back. But what you do get in glycolysis is that you do have those electron acceptors, those NAD pluses. You grab 2 sets of electrons and 2 hydrogens off the glucose. You take 2 NAD pluses, and they accept it. As you break the bonds here, you are, when you put the glucose, when the glucose is run through the dehydrogenase enzymes, and you're breaking bonds to release energy for the ATP, the NAD plus will grab the electrons and hydrogens that get released, and they now are carriers. They're carrying those electrons. They are reduced. They have picked up the electrons. They are reduced. They're carrying the hydrogens and the electrons. So you have 2 NADHs plus extra hydrogens that are still attached to it. Stripped it right out of the glucose. Right as we're ripping it in half, it strips those right out of there. And that's a lot more important than what you just made. You also make 4 ATP, because that substrate level phosphorylation takes the glucose, shoves it into the active site of the enzyme, strips out the electrons and the hydrogens, transfers that energy, that energy that's getting released to the ATP, and hey, we make 4 ATP molecules. But it's more important that the two NADHs are holding on to high energy electrons. Now, something you need to remember. You make 4 ATPs in glycolysis, but that's the total. There is only a net gain of 2 because you had to start the process of glycolysis and you had to burn 2 ATPs to start the process. So you burn 2 ATPs and you make 4. I mean, so you're still up by 2 ATPs. So you burn 14.6 kilocalories of energy. That's 7.3 each, 2 ATPs. An ATP is 7.3 kilocalories. So you burned 14.6 kilocalories and you made 29 29.2. You stored 29.2 kilocalories. Yeah. So you burned 14 kilocalories and you stored 29. So you're ahead because you got 4 ATP out of it. So you're ahead by two ATPs. You burned 2 ATPs, you made 4. Don't get confused. You know, a test question would ask you, what is the total number of ATPs made during glycolysis? The answer is 4. What is the net total? The answer is 2. Now, why do we care about this? Why am I even worrying about this? Because this involves the practical application is this is what happens when an individual depletes the oxygen in their myoglobin. They deplete the oxygen in their myoglobin and they go into oxygen debt and they go into lactic acid formation and their muscles hurt. This is why someone who has run a marathon is going to have very sore muscles because they've used up all their oxygen. They're running off of glycolysis and not electron transport. So they're running off of glycolysis. Their muscle cells will function, but the pyruvic acid doesn't have any oxygen to let it go into, pyruvic acid won't go into the mitochondria unless there's oxygen in the muscle cells. And if the muscle cells have depleted their oxygen, that pyruvic acid is going to become lactic acid. That's going to make the muscles sore. It's also going to cause them to breathe faster because lactic acid lowers blood pH. So here we have a situation where someone has run a marathon. They're breathing rapidly. Why are they breathing rapidly? Because of their low blood pH caused by the lactic acid, caused by the buildup of lactic acid in the skeletal muscles. Are they getting energy? Yeah, they're getting a little energy. They're getting 4 ATPs instead of 36, but they're getting energy to function. Very long duration strenuous activity will usually deplete the oxygen in the muscles, and that'll be oxygen debt. It will cause the muscles to hurt because of the accumulation of lactic acid. Lactic acid will cause an increase in the breathing rate because lactic acid lowers the blood pH. The medulla oblongata in the brain thinks, oh, pH is low, therefore we have too much CO2, we better be breathing faster. So that's why it's important to understand what happens in an individual. So this is this slide right here, a very good source of information. Glycolysis takes glucose and breaks it down to 2 pyruvic acid molecules that will go into the Krebs cycle. If there's no oxygen available in the cells, then it becomes lactic acid. We make 2 NADH molecules. Each one holds 2 high energy electrons. 4 ATPs were made, but we had to burn 2. We had to start by burning 2 to get this. We had to use 2 ATPs to start the process, so our net gain is only 2. We burn 2 to make 4. And so we can run on glycolysis as long as there's carbohydrates present, as long as there's glucose present, and we are always going to have glucose. You know, we don't deplete our glucose. Remember that we can go for a month without food. So we've got vast amounts of reserves of glucose. We don't lose glucose to the point where we burned up all of our glucose. It takes a while to do that. So running a marathon is not going to deplete your glucose. It will deplete your stored oxygen. So now, glycolysis is not very efficient. You know, what, 4 ATPs out of a possibility of 36? That's not, you know, very efficient. That is 11% efficient, you know, in producing ATPs. It's not very good. Cellular respiration, on the other hand, is very efficient. We use oxygen here. Cellular respiration involves the use of oxygen. It is an aerobic process. And we use oxygen to grab the electrons off of the glucose molecules, or rather the pyruvic acid molecules, because we've broken remember we've split the glucose into half now, into two halves. It's pyruvate on each side. We have two pyruvates. And we oxidize those pyruvates, and we get a lot of energy coming off here. Now, ATP, we store energy in the ATP. You know, the ATP molecule will temporarily transfer a high energy phosphate to something, like, for example, the sodium potassium pump to power the pump. You know, it'll transfer the energy. It'll send its third phosphate over to the pump, and the pump will be powered. It will send the third phosphate to a muscle fiber in our skeletal muscle so it can contract. We'll see all about that when we get into the muscle chapter. So we can use the ATP is the currency that we pay the bills with in our cells. We use the ATP to transfer energy to the structures in our bodies that need energy to work, like the contraction of muscle cells, like the sodium potassium pump, like sending nerve impulses in here. And so the ATP will readily give up its third phosphate with its attached energy to get things done. That's what it does. Now, and we make ATP by phosphorylation. It doesn't matter whether it's substrate level or oxidative. We make ATP by forcing a third phosphate onto the adenosine diphosphate, using energy to force that third phosphate in there, making a high energy bond. We make the high energy bond, holds the phosphate on there. Now we have ATP, but ATP only lasts for 7 seconds. This is ATP right here. You know, there's the adenine, there's the ribose sugar, and here are the 3 phosphates. It is the third phosphate held on by a high energy bond that we grab the energy off of glucose to put that bond back together again. And ADP and inorganic phosphate go back and forth all the time. They're not destroyed in the reaction. ATP gives up that third phosphate temporarily to do something. And then that low energy phosphate now is reattached with energy to the ATP. And now it can go somewhere for 7 seconds. And it goes back and forth, back and forth. You know, low energy, high energy, low energy, high energy. When we release that third phosphate, we're going to drag the high energy with it to do something, to pump the sodium potassium pump, to contract a muscle, to send a nerve impulse. So glycolysis takes place in the cytoplasm. It gives you 4 ATPs total. It also has captured 4 high energy electrons. You got 2 NADHs. They're carrying high energy electrons. They strip those off of the glucose when it's split. You know, the dehydrogenase enzymes stripped off those electrons, pulled off the hydrogens. That's great. And in the process of breaking those covalent bonds, we were able to power up 4 ATPs. That's fine. However, we want more. We want a lot more. If we're going to assume that we are in an oxygen-rich environment and everything else takes place in the mitochondria, the pyruvic acid that got produced by glycolysis will make its way into the mitochondria. The mitochondria, you know, the energy conversion structures are in the cytoplasm. Glycolysis took place in the cytoplasm. The pyruvic acid leaves the cytoplasm and goes into these mitochondria. And it goes into the matrix of the mitochondria. This is the matrix. It's the area, you know, in the middle of the mitochondria. You know, there's the inner membrane there. These little folds are called cristae. And in these spaces between the folds is the matrix. And that's where the Krebs cycle takes place. This only happens, though, if oxygen is available. If there's no oxygen, we're not even going to get near the mitochondria. So the pyruvate molecules that got made, we just split the glucose in half. Each pyruvate will go into the matrix. Each pyruvate will immediately be oxidized when it goes in here into the mitochondria, and it will be oxidized, and a carbon will be stripped off. Carbon dioxide, the carbon dioxide that we breathe out, forms inside the only place that it forms is in the mitochondria of our cells, and only in the matrix. When the pyruvic acid goes into the matrix of the mitochondria, it is immediately oxidized. The 3-carbon pyruvate is immediately oxidized to 2 carbons. The 3rd carbon combines with oxygen and goes off as CO2. That's our first molecule of CO2. The 2-carbon segment left over combines with an enzyme, a coenzyme, to form a substance called acetyl-CoA. And the electrons that were released when we oxidized the pyruvic acid are captured by NAD+. So pyruvic acid a molecule goes into the matrix of the mitochondria. It's immediately oxidized from 3 carbons to 2. The 3rd carbon comes off as carbon dioxide. The electrons that were released are captured by the NAD+. We get 2 more NADHs in here. The 2-carbon compound is oxidized with coenzyme A to form something known as acetyl-CoA. So this is what happens. There's your pyruvic acid right there. Goes in the mitochondria. We're in the matrix of the mitochondria. It is immediately decarboxylated. The carbon is ripped off. There goes the first CO2. Then the remaining 2-carbon structure is oxidized, and we get 2 NADHs. Coenzyme A comes along and oxidizes the 2-carbon compound and gives us something called acetyl-CoA. What do you need to remember about this? Well, pyruvic acid is oxidized, and we get a carbon dioxide coming off. We also get two NADHs. The end result of this is acetyl-CoA or acetyl-CoA, and that goes into the Krebs cycle. The Krebs cycle, all the Krebs cycle does is take the acetyl-CoA and combine it with a 4-carbon molecule and produce citric acid. So, and this is a stepwise progression to capture a lot of high energy electrons. The Krebs cycle is all about capturing high energy electrons. That's all it does. That's its primary role. It'll make a couple ATPs for you. But what happens is the 2-carbon compound, the acetyl-CoA, enters the Krebs cycle, combines with a 4-carbon molecule called oxaloacetate. You don't need to remember that, forming citric acid, which then gets oxidized several times. There goes your pyruvate. It's being oxidized once to get CO2. We make our acetyl-CoA, and this is going too fast. Here we go. This is the Krebs cycle. And I'll tell you, the Krebs cycle is something that you relearn every time you look at it. There was a time when I was in graduate school and I could sit down and write out every step from memory with all the reactions going on and explain what they were doing. And, you never really use that again. But here's what's happening. Here comes the pyruvic acid. There goes your first CO2. Decarboxylase enzyme is ripping off the first carbon. We oxidize what's left. We grab a couple of NADHs and high energy electrons, and that's more important than anything else. The acetyl-CoA formed when we react, when we oxidize it with coenzyme A. There's our citric acid. That's why we also call it the citric acid cycle. It's oxidized here. Another CO2 comes off. More NADHs come off. We oxidize it again. More CO2 comes off. We get more high energy electrons. We make 1 ATP. We grab more high energy electrons by oxidizing with FAD. We do it one more time over here. And we end up with a 4-carbon compound again, ready to go again. It's just all the Krebs cycle is a sequential process of generating high energy electrons and the formation of CO2. Now there are 2 pyruvates that go in here. So everything you see here is double. Let's skip to the bottom line. Bottom line of the Krebs cycle. This is what you need to remember. You get a grand total of 8 NADHs. Each one of those has 2 high energy electrons. You get 2 FADH2s. Each one of those has 2 high energy electrons. Where'd they come from? From the breakdown, from the oxidation of the pyruvic acid and the compound that is made into the citric acid and so on. So you get 8 NADHs and 2 FADH2s. You get 6 CO2s and you get 2 ATPs. 6 CO2s. Remember the formula? C6H12O6 in the presence of 6 oxygens yields 6 CO2s plus 12 waters and so on, or 6 waters. And release of a lot of energy, or the capture of a lot of energy. Well, here we've got 2 more ATPs. That's not a lot of energy. Our net so far, our net amount of ATP, we burn 2 to make 4. That gives us a net of 2 in glycolysis. Now we have a net of 2 here. So far we've managed to generate 4 net ATP. That's not a lot. More importantly, these 8 NADHs and 2 FADH2s, that's where we parked the high energy electrons and their hydrogens. So this is the Krebs cycle. Everything else is sort of extraneous. This is what's important. And so why do we care? Well, This is where CO2 is coming from in our cells. It's from the Krebs cycle inside the mitochondria. All the CO2 that we release, all the CO2 that is affecting our blood pH and our breathing rate. Because, you know, when blood pH goes down and we become more acidic, the medulla says, breathe faster to blow off CO2 because we're too acidic, because CO2 combines with water and plasma to form carbonic acid, and that makes us more acidic. So the CO2 that's coming off is coming off at the Krebs cycle. And the faster we burn glucose, the more CO2 we generate, the more acidic our blood gets. And if we don't have oxygen present, we don't get into the Krebs cycle. If we don't have oxygen present, we form lactic acid. And guess what? We still breathe fast because the lactic acid makes our blood pH low. So the practical side of this, this is where our CO2 comes from. Yes, we've got lots of high energy electrons, and those are going to be very important to us here in a minute. But from a practical standpoint, the reason that we breathe rapidly after exercise is not to get more oxygen. We've got tons of oxygen in our blood and in our cells. We just don't have, we got lots of oxygen, but we're also generating a lot of CO2. Like it's the CO2 that makes our blood acidic, and the medulla thinks we got to get rid of it. So we breathe fast to get rid of the CO2 that's coming right out of the matrix of our mitochondria. Now the electron transport chain is the last step. Electron transport chain and oxidative phosphorylation. This is where oxygen finally gets used. We've used no oxygen up to this point. Glycolysis doesn't use oxygen. Krebs cycle doesn't use oxygen. Krebs cycle won't run if there's no oxygen present. We'll never get out of glycolysis. We'll make lactic acid instead. Within our mitochondria is a series of enzymes embedded in the inner membrane. These are the electron transport enzymes, and they will physically move electrons from one enzyme to another. These are high energy electrons. And these electron transport enzymes function as pumps. They're going to pump hydrogens, the hydrogens that are attached to the NADs. The hydrogens that are attached to the NADs, along with those high energy electrons, those electrons are going to be used to pump those hydrogens into the membrane space between the inner and the outer membrane. We see these are all enzymes. There are essentially three big enzymes in here that are going to be responsible for be the electron transport chain. So, okay. And before we start this, I'm going to have to wrap it up for today because I don't have enough time to get into it. I've probably thrown more at you than I should have. But we will stop here. And on Tuesday, we will pick up with this and I'll review it a little more. And we're going to finish this chapter on Tuesday, and we're going to start something a whole lot more interesting. We're going to look at the physiology of bones. So anyway, if anybody has any questions on this, shoot me an email. I'll be glad to answer it for you. And this is probably the most challenging content that you're going to encounter. It gets a whole lot easier after this. But again, I'm trying to show you the practical application of this, because I'll tell you, no one's going to run up to you and say, hey, how many ATPs do you think I'm getting off my electron transport chain? So it's never happened to me anyway. So, okay. I'm going to get us out of here, and there is no lab this afternoon. There are no labs this week because of, fall break sucked up 2 lab days. So don't expect to come to lab this afternoon.